So in this session, we'll go through the chromosome structure, karyotyping and barr body. You can see here, chromosome means colored body. These are deeply stained rod-like structures within the nucleus, seen during cell division, especially the metaphase. So during interphase, which is the resting phase, nucleus contains network of fibers called chromatin threads, or it is in the form of threads or granules. The uncoiled portion of the chromosome is the euchromatin, which is genetically active, and the coiled portion of chromosome is the heterochromatin, which is genetically inert. So during cell division, the chromatin threads are tightly coiled, so the individual chromosomes are readily visible. So difference between euchromatin and heterochromatin. Euchromatin is lightly stained chromatin, whereas heterochromatin is darkly stained chromatin. So there will be less coiling here, there will be more coiling here. So euchromatin is the active chromatin, heterochromatin is the inactive chromatin. This is expressed during interphase. This does not express during interphase. So contains structural genes, contains repetitive DNA sequences. So mutations occurs frequently in euchromatin because it is active, and mutations occur less frequently in heterochromatin. So contains guanine and cytosine bases predominantly, contains adenine and thymine bases predominantly. So types of heterochromatin. Facultative heterochromatin, initially active but later becomes permanently inactive. Example, inactive X chromosome barr body. Constitutive heterochromatin, so inactive throughout the life of an organism, seen at primary and secondary constructions of chromosomes. You can see this is an ultramicroscopic view of the chromosome. You can see these are the sister chromatids. This is the centromere. You can see this is the short arm and this is the long arm. This is the P arm and this is the Q arm. So humans have 46 chromosomes. Haploid number is 23, diploid number is 46. 46 XY male, 46 X female. 22 pairs of autosomes and 2 sex chromosomes, X and Y, or X and X. So this is the centromere or kinetochore. So you can see the spindle being attached here. These are the sister chromatids. So parts of a chromosome, chromatid. So in metaphase, chromosome has two symmetrical structures called sister chromatids, centromere or kinetochore. Both chromatids are attached at centromere. Human chromosomes are monocentric, only one centromere. Mitotic spindle attaches to centromere. Telomere, free ends of sister chromatids are called telomeres. Prevent fusion of the chromosome with each other. Deletion of telomere makes the ends sticky. Secondary constriction, the narrowed portion of the chromosome other than centromere is a secondary constriction. Nuclear organizers, so regions of secondary constriction with genes that code for ribosomal RNA. Satellite, round elongated knob-like appendage of chromosome seen in acrocentric chromosomes only, connected to rest of the chromosome by thin filament of chromatin. Microsatellite, short sequences of DNA repeats 2 to 6 base pairs long. So parts of the chromosome, P arm is a short arm, P for petit small. Q arm is a long arm, Q is next to alphabet P. So there is no specific name for it. And see, in this we'll get well versed with the parts of the chromosome as well as how we read or the nomenclature of the chromosomes. You can see here, this is the telomere of the P arm, this is the telomere of the Q arm. Q arm is the long arm, P arm is the short arm. These are the two identical sister chromatids. You can see here, this 1, 2, 3 are the regions. Here within a region there are bands, and within a band there are subbands or locus. Suppose if I write 3p22.1, so 3 stands for the chromosome 3, P stands for whether it is the short arm or long arm, and then 2 stands for, so in the short arm the second region, and the next 2 stands for in the second region the second band, and next 1, this stands for subband or the locus. So between the band and subband we need to keep a dot. This is how the chromosomal nomenclature is read. Chemical composition of chromosomes, mainly made up of DNA, histone proteins and non-histone proteins. Histone proteins are H1, H2A, H2B, H3 and H4. So important for repression of genes, maintain integrity of chromosomes. Non-histone proteins, actin, myosin, alpha and beta tubulins, helps in chromosome condensation, DNA replication and transcription. You can see the coiling of chromosomes. This is the primary coiling where it appears like DNA. So then the DNA is coiling around the nucleosome. You can see here, other than H1, remaining all histone proteins, they are forming an octamer called nucleosome. So around which there is coiling of the DNA. So this is secondary coiling, euchromatin. And you can see here, these are solenoids, like 6 nucleosomes per bend. So here there is tertiary coiling where this Coiling results in formation of heterochromatin, and ultimately there is supercoiling or quaternary coiling, which results in the formation of the chromosome. Primary, secondary, tertiary, and quaternary coiling. So H1 does not take part in nucleosome formation. H1 is linker histone protein. Then morphological types, you can see here. Metacentric, where centromere is at the center and both arms are almost equal. Then submetacentric, whereas centromere is slightly away from the center. There is a short arm and there is a long arm. And acrocentric, where you can see the satellite also, there is a short arm and there is a long arm. The centromere is located more towards the end. And telocentric, where centromere is at the end. There is no short arm, there is only long arm. So telocentric chromosomes are absent in humans. Again, same thing, metacentric, submetacentric, and acrocentric with the satellites. So satellites are seen only in the acrocentric chromosomes. So karyotype and karyotyping, the chromosome constitution of an individual, which is known as the karyotype, and the process of obtaining the karyotype of a cell is called karyotyping. So if you want to elaborate this definition, karyotyping is the process by which photographs of chromosomes are taken in order to determine the chromosome complement of an individual, including the number of chromosomes and any abnormalities. So procedure, collection of sample, blood, amniotic fluid, chorionic sample, fetal blood. Planting, so planting in a culture medium, so especially RPMI 1640 or TC199. Then fetal bovine serum is added for nutrition, and phytohemoglobin as mitotic agent to enhance the solution. And then we can also add antibiotics to prevent bacterial growth. So incubation, 37 degrees centigrade for 72 hours. Then colchicine block cells in metaphase. Harvesting, cells washed with hypotonic solution or kept in hypotonic solution for 20 minutes, sodium citrate, which results in swelling of cells. Then the cells are mixed with glacial acetic acid and methanol. In slide preparation, cells are dropped onto a slide. Then we can do the GM staining, which is more in favor of the G banding. So there will be pre-treatment with trypsin to partially digest the histone proteins before staining. So GM staining results in G banding. So again, as a flowchart, sample collection, culture setup, incubation, colchicine block, harvesting, slide preparation, banding and staining, and karyotyping. So you can see here same thing. Blood sample is being collected. So phytohemoglobin to the culture media. So culture at 37 degrees, incubation, then colchicine block, then hypotonic saline, cells fixed. So spread on the slide, then stain, and we'll get a picture, and then separate all the chromosomes and arrange them in lengthwise, so which gives us the karyotype. You can see this is a picture obtained after the culture and fixing and slide preparation and staining. You can see the banding also here. So now the chromosomes are arranged as per their lengthwise. You can see here 1, 2, 3, 4, 5, 6, 7, 8, all 22 autosomes X and Y, allosomes. Now if you observe, the first, second, third chromosomes belong to group A as per Denver's classification. So in that, first and third are metacentric and second is a submetacentric. Let us see that. See here, first and third are metacentric, second is a submetacentric. And here you can see 4, 5 belong to the B group, where you can see both are submetacentric, and here all are submetacentric. Here all are submetacentric. Even X chromosome is also submetacentric here. So then 13, 14, 15. 13, 14, 15. 21 and 22. All these are acrocentric. All these are acrocentric. You can see here 13 to 15 acrocentric. Then 16 to 18. 16 is a metacentric, whereas 17, 18 is submetacentric. Let us see here. 16. 16 is metacentric. 17, 18 are submetacentric. And you can see here 19, 20 are metacentric. You can see 19, 20 are metacentric. Then 21, 22 and Y are acrocentric chromosomes. So this is the Denver's classification, A, B, C, D, E, F, G. So 1, 2, 3 in A, 4 and 5 in B, 6 to 12 in C along with the X chromosome. So D is having 13, 14, 15, E is having 16, 17, 18, F is having 19, 20, G is 21, 22 and Y. So X belongs to C and Y belongs to G. So various other bandings like Q banding, where we use quinacrine, a fluorescent dye. It is useful for studying heterochromatin of chromosomes 1, 9 and 16. Then R banding, reverse banding, where the slides are preheated before GM staining, which will produce reverse G banding pattern. Then C banding, so where chromosomes are denatured with acid or alkali or heat prior to GM staining. It is useful to study of constitutive heterochromatin, dark banding, and there is NOR banding to study the nuclear organizing regions. So G banding, Q banding, R banding, C banding, and NOR banding. So fluorescent in situ hybridization is another technique. So this method is based on unique ability of a portion of single-stranded DNA to anneal with its complementary target sequence on a metaphase chromosome, interphase nucleus, or extended chromatin fiber. So it is mainly based on the capacity of a single-stranded DNA to attach with the complementary sequence on the chromosome. So what happens here is, we'll prepare a DNA probe which is labeled with a fluorochrome. Then after hybridization of this probe with the patient sample, then we will take the pictures using the fluorescent microscope. So this fluorescent in situ hybridization is widely used for clinical diagnostic procedures alongside with the karyotyping. You can see here, this is more or less like karyotyping only, but we are using a fluorescent technique. Here you can see these are the target areas which we wanted to identify. So this is a photomicrograph which is obtained from the slide. So now barr body, it's also called as sex chromatin or X chromatin. Barr and Buttram in 1949 observed it in nuclei of phrenic nerve cells of female cat. Highly coiled, genetically inactive chromosome, genetically inactive X chromosome. So it is subperipheral below the nuclear membrane. So it is of different shapes like plano-convex, drumstick, or barbed wire. You can see the barr body in case of female. In male, it is not there. You can see here the barr body, and you can also see your drumstick body in neutrophil, barr body in somatic cell. So from buccal smear also, so buccal smear, scraping from buccal mucosa, and that will be taken onto a slide, smeared, then evenly spread, fixed with alcohol and stained with thionin. We can observe the barr bodies. So lyons hypothesis, in early embryogenesis, so one of the two X chromosomes becomes inactive and forms barr body. So process of X chain activation is initiated by ZIST gene, X inactive specific transcript gene, located on long arm of X chromosome. Located on long arm of X chromosome. Located on long arm of X chromosome. So DNA methylation is the main step in the inactivation of X chromosome. Why it is being done? It is for dosage compensation. That is, X-linked protein product should be equal in both males and females for normal phenotype. So what is actually happening here is, either paternal or maternal X is inactivated randomly in different cells in females, because inactivation occurs on 15th day at 5000 cell stage. So what happens once either paternal X or maternal X is decided in this 5000 cells, then whatever cells which develop from that particular cell will have the same X chromosome inactivated. Suppose if I take one cell from this and that cell has maternal X inactivated, and whatever cells which develop from that particular cell will have same maternal X inactivated in them. So that is why in females normally there will be two cell lines. So presence of two or more populations of cells with different genotypes in an individual developed from single fertilized eggs is mosaicism. The same thing, presence of two or more populations of cells with different genotypes in an individual developed from two or more fertilized eggs is called chimera. So point to remember is, once inactivated, the same X chromosome remains inactivated in daughter cells derived from cell after inactivation. So few genes and short arm of inactivated X chromosomes are not inactivated. They escape inactivation. So how to calculate number of barr bodies? Number of X chromosome minus 1. So in normal male, no barr body. In normal female, one barr body. In Turner syndrome, as there is only one X, there will be no barr body. In Klinefelter syndrome, there will be one barr body. Thank you.